The Process of Mitosis by Sofia Antolinez and Martina Angueira. Here we can see different elements that will be used during our representation. You can see the chromosomes, which are represented in purple and red. You can see the nuclear envelope, which is represented in green. You can see the cell membrane, which is represented in blue. And you can see the polar fibers, which are represented in yellow.
Mitosis is a process of cell division, but before it begins, interphase must happen. It is divided into three different steps, which are G1 phase, S phase, and G2 phase. The G1 phase is when the cell begins to create more proteins so that it can grow, preparing for the later division it will go through. The S phase is when the DNA gets synthesized, which means that it gets coiled up. In the G2 phase, the chromosomes replicate and form identical chromosomes named chromatids. By the end of the interphase, the cell will get double the number of chromosomes, going from 46 to 92 chromosomes.
After the interphase comes the prophase. In this stage of the mitosis, the chromatids remain attached and meet at the centromere. The chromatins then condense into discrete chromosomes. This condensing process happens until the cell reaches metaphase. The nuclear membrane, also known as the nuclear envelope, which refers to the double lipid bilayer that surrounds the genetic material and the nucleus, breaks down. The spindles also begin to form when the centriole moves to opposite poles and the microtubes begin to polymerize. This happens in the cytoplasm. This is the first stage of prophase.
So after prophase, we have the late prophase, and here the nuclear envelope is breaking up. Polar fibers reach from the cell's poles, each towards the center. Chromosomes begin to migrate towards the cell center. Here is a visual representation of late prophase. So after prophase, we have metaphase. Here, the spindle finishes developing and the chromosomes align at the metaphase plate. While this is happening, the nuclear membrane is completely fragmented, and the microtubes that are composing the spindle fibers extend from the poles up to the center of the cell. Here, you can see what's going on in metaphase. So the chromosomes have been aligned at the metaphase plate, which is this one. We can also see how there is no nucleus anymore. It has been fragmented. And finally, we can see how the spindle has been finished its development.
After the metaphase comes the anaphase. This is when the sister chromatids that are paired up as chromosomes in the metaphase plate are separated and they begin to move towards the separate poles or ends of the cell. During this step, it is important to highlight that spindle fibers that are not paired to any chromatids begin to lengthen to further elongate the cell. When anaphase ends, each of the opposite poles will contain a complete set of 46 chromosomes. The two poles move further away during the course of anaphase. So in this visual representation, we can see that the spindle fibers are connected to different set of chromatids, and the chromatids are being moved away from the center of the cell to the separate poles each. We can also see that there are some spindle fibers that are not connected to any chromatid, and they are elongating the cell by lengthening even more.
After anaphase comes telophase. Here, the chromosomes are cordoned off in order to make the new nuclei of their respective daughter cells that are being created. The polar fibers are still lengthening, the nuclei are starting to form at the opposite poles. And once its changes are completed, most of mitosis is now complete. because the genetic contents of the cell have been duplicated and divided. You can see it's happening here. The fiber poles are still here and they're still lengthening. You can also see the nuclei forming through two and its respective chromosomes that have been now duplicated. The last step after telophase is called cytokinesis. This is the last step, and it is when the cell actually divides. Here, you can see the two cells that have been now divided with its respective nucleus, chromosomes, and centriole. As you can see, these two cells that have now been duplicated are now going to begin interphase so that the cell cycle can continue.